Hi guys, today we're going to talk about two different mediums, Loeffler's medium and Tellurite Agar, which is also called potassium Tellurite Agar or cysteine Tellurite blood agar. Both these medias are used to grow C. difficile or Clostridium difficile. I'm going to start by giving you a scenario or a question where information about these media is crucial to answer the question. Okay, so just imagine there is a outbreak of a respiratory disease in a very rural community somewhere in Pakistan or somewhere in India where vaccination rates are not very good. Um, and what they tell you is that the bacteria are identified to grow. They grow in a very specialized culture that may contain sheep's blood, cysteine, and potassium telluride. And the question stem is emphasizing that once placed in an agar like this, the colonies that grow, they're black and they have a very shiny surface. Okay. Also, there could be a question that says that um, the bacteria grown in Loeffler's media with that, or they may not even mention Loeffler's. They may just say it grows in a medium containing serum, glucose, and a nutrient broth. And, the bacteria are club-shaped, and they see metachromatic granules intracellularly in these club-shaped bacteria. So what would the answer be? Obviously, it would be C, Corynebacterium diphtheria. So now let's get into the medium. So Loeffler's medium is an enrichment medium. It is used for both the cultivation of Corynebacterium diphtheria, which is a diphtheria-causing bacteria. The medium is made up of serum, glucose, and a nutrient broth. It encourages the production of metachromatic granules. These bluish green granules, which you can see in the picture right on top in these bacteria, and they can be seen under the microscope using special stains. Now, here's the catch. The Loeffler's medium is not selective, means that it can make other bacteria grow as well, but it enhances the growth of C. diff by providing ideal conditions for the bacteria to thrive and express the characteristic metachromatic granules. So, it is basically used to see, to observe microscopically, C. diff, to observe the morphology and the granule formation. Okay, moving on, let's look at Tellurite agar. So it's also called a potassium Tellurite agar or cysteine Tellurite blood agar. So remember, in the question stem, you'll see potassium. It could be confusing. They might not say tellurite, they might say potassium tellurite. They may even mention cysteine. They may mention blood in the agar with cysteine and potassium. Okay, so what is the purpose of this medium? It's a very important medium because it isolates this bacteria. It selectively and differentially isolates this Corynebacterium diphtheria by allowing it to grow selectively. It contains tellurite salts, which inhibit the growth of most other bacteria, and it may contain blood or cysteine for enriched growth. What happens is that C. diff, it reduces the tellurite salts to a metallic tellurium, producing distinctive black or gray colonies. So these colonies are going to be black or gray. It's going to be very shiny black or gray, like a starry sky, like a black sky. Um, it inhibits non-diphtheroid bacteria, so it isolates C. diff. So this is very important. This is a definitive diagnostic, like it would 100% confirm the presence of this bacteria. It is used for initial isolation and presumptive identification of C. diff in clinical samples. So, since you're talking about the two different media, let's also look into the organism itself. We all know that the C. diff is a gram-positive bacillus. It is transmitted via respiratory droplets, um, and it causes the childhood illness diphtheria. The Obviously, this infection is more common in areas where vaccination is not done very effectively or not done at all, and it can spread rapidly in under-vaccinated areas of the world, such as rural areas like Pakistan, India, lots of other rural places. Um, The cultures should be collected, when cultures are collected, they're collected prior to the administration of antibiotics. And obviously, the growth of these organisms are the media that are specifically chosen for the growth of this organism is cysteine tellurite agar, Loeffler's medium. Um, Cysteine tellurite agar, it enables the growth of pathogenic Corynebacterium species while suppressing the growth of other diphtheroids. And it makes black shiny colonies. And that is the end of this presentation. Thank you.